Okay. So these are the topics that will be taken in the course of advanced cell biology. We'll start today with biomembranes. In next lecture, we'll talk about the transmembrane transport. And the main reference for the course is Molecular Cell Biology, 8th edition, 2016, by one of the famous scientists, Lodish et al. So what is what is the plasma membrane? Actually, cells are partitioned into smaller compartments, which are the organelles. And each organelle is surrounded by biomembranes. So each organelle contains a unique group of proteins, and actually these proteins specifies the function of each organelle. So in other words, you have a mitochondria, you have a Golgi apparatus, and these are all our organelles. Each different organelle have a specific set of protein that specify its function. So this protein enable organelles to carry out its characteristic cellular function. The cytosol also contain its own distinctive proteins. So actually the cytosol, it is a part of the cell where the organelle exists inside it. So biomembrane actually form closed structures based on a similar structure as the bilayer, the lipid bilayer. It controls the movement of molecules between the inside and the outside of the cell, and as well control the movement into and outside of the organelles. While the cytoskeleton, actually it is a dense network of protein filaments that is found in the cytosol and mechanically support cellular membranes. Each type of cell exhibits a unique design defined by the shape of the cell, which is the cytoskeleton, and the location of its organelles. And these cytoskeletal proteins are among the most abundant protein in a cell. And you can understand that we have different types of cell inside our body that forming tissue. And you can see that, and you can understand that as well, that each type of cell have a different shape, different dimensions. The different shape and dimension owing to the presence of the architecture of these cytoskeletal proteins. From this figure, you can see that actually most of the area is occupied of the cell is occupied by cytoskeleton, then the internal membranes, and then the plasma membrane. So you can understand now there is abundance of cytoskeletal protein or cytoskeletal elements, and it is the most abundant element inside a cell. So in this section, we will consider the basic structure of biomembranes. Actually, the first component is the lipid component of membranes. It affects the shape of the membrane, affect the function, and play a very important role in anchoring proteins to the membrane. It modifies membrane protein activities, and as well it transduces a signal to the cytoplasm. So the lipid in the membrane transduces the signal coming from the outside the cell into the cytoplasm. We will then consider the general structure of membrane protein. So as you know, the two main constituents of the biomembranes is lipids and proteins. The unique function of each membrane is determined largely by the group of protein within and adjacent to that membrane. So let's talk first about the lipid composition and structure organization. Lipids of the plasma membrane actually in the form of a sheet-like of a phospholipid bilayer. It's a 2-molecule thick. You can see it's a 2-molecule thick. So actually this is a section of a small part of the plasma membrane. And you can see it's a 2-molecule thick because you have 1 molecule on top, 1 molecule on bottom, and they are oriented opposite to each other. The hydrophobic core is formed by the hydrocarbon chains, as you can see, while the polar heads are oriented to the outside. If you look under the electron microscope, when these section or specimen are stained by its specific stain, which is the osmium tetroxide, this osmium tetroxide, because it's a polar chemical, it binds tightly with the polar heads. That's why it appears on the EM as a railroad track appearance, because actually it stains only the heads, because the stain is polar and it stains the heads. And you can see that the thick of the layer is actually 3 to 4 nanometer thick. So the lipid bilayer has two important functions or properties. First, the hydrophobic core, which is formed of the hydrocarbon chains, is an impermeable barrier. Prevent the diffusion of water soluble, which is actually the hydrophilic solids. While the second property of the bilayer, it's very stability. It's very stable. This bilayer structure is maintained by hydrophobic and van der Waals interaction between the lipid chains. That's why it looks always aligned, the lipid molecules looks aligned. Even though the exterior aqueous environment can vary widely in ionic strength and pH, the bilayer has the strength to retain its characteristic architecture, because this actually is crucial. Otherwise, the biomembrane will be destructed and the cells will be destroyed. So there are three classes of lipids are found in the biomembrane. You have the phosphoglycerides, you have the sphingolipids, and you have the steroids, which majorly the cholesterol. All these three classes of lipids are amphipathic molecules. What's the meaning of amphipathic molecules? That each molecule have a part that is hydrophobic and a part that is hydrophilic. The three types of, or the three classes of these lipids, they differ in the chemical structure, they differ in abundance, and they differ in function as well, what they do in the membrane. So what about the phosphoglyceride? Actually, it is the most abundant type of lipid in the biomembrane, and it is derivatives of the glycerol 3-phosphate and are classified according to the nature of its head. If you remember, the nature of its polar part, the nature of its the part of the molecule which is to the exterior in the biomembrane. So you can see here PI, which is the phosphatidylinositol, is a class of phosphoglyceride. So initially we have three types of lipid: phosphoglycerides, sphingolipid, and steroids. And now we are talking about phosphoglycerides. And now we are talking about phosphatidylinositol, which is a class of phosphoglyceride. You can see in this one, in which the head group consists of a sugar derivatives. So that's the head group. Sugar derivative, which is the inositol ring, that is linked to the hydrophobic fatty acyl tail. You can see these all are the fatty acyl tail by a phosphate group. Here is the phosphate group. And that's why it's called phospholipids, because you always find the phosphate that is linking the polar head, with the non-polar or hydrophilic, with the hydrophobic tail, which is actually hydrocarbon chain. So what about the protein composition of the biomembrane? Although every biomembrane has So for the protein composition, although every biomembrane has the same basic layer structure, so actually you need to understand that biomembrane have the same phospholipid bilayer. So the main player which affect the function of the biomembrane, actually the type of protein associated with the membrane. So the protein associated with the membrane are responsible for its unique activity. And the density and types of these protein may vary depending on the cell type and the subcellular location. For example, the inner mitochondrial membrane is around 76% protein content, while the myelin sheath or the myelin membrane of the neuron is only 18%, and most of it is lipid. The high phospholipid content of the myelin in the neuron allows to the electrical impulse to be insulated while propagating inside the nerve. And these actually very good in maintaining the signal of the impulse throughout the nerve and separating it from the external environment. Some proteins, going back to proteins, some proteins are buried within the lipid layer, lipid-rich layer. Other proteins are associated, as you can see from the figure, by two sides. One is exoplasmic, which is outside the cell, and one is cytoplasmic, which is inside the cell. And usually they are connected with a channel or a transmembrane part of the protein. Usually the external surface or the exoplasmic part of the protein usually bind to the signaling proteins or ions or small metabolites comes from the outside. And they bind as well to adhesion molecules on other cells in case these type of cell need to be anchored to another cell of the same type to form the tissue. While the cytosolic side of the protein usually used for anchoring the cytoskeleton to the membrane and as well in conveying the signal from the outside to the inside of in the cytoplasm through a signaling pathway. And as well the connecting part, which is transversing the phospholipid bilayer, usually it's either channel, pores, or even a solid part of the protein. So actually proteins have three different types and got attached to the membrane in three different ways. And basically the nomenclature of these three different proteins is based on the way they are attached to the membrane. So membrane protein can be classified into three categories: integral, lipid anchored, and peripheral, on the basis and the nature of the membrane protein interactions. For the integral membrane protein, which can be called the transmembrane proteins, they cross the phospholipid bilayer. They build of three segments, as we have just seen in the previous slide. Most transmembrane proteins are glycosylated. What's the meaning of glycosylated? That's mean they are attached with a moiety of carbohydrates with a complex branched sugar group. While for the lipid-anchored membrane proteins, they bound covalently to one or more lipid molecules. So you can find that some proteins in the biomembrane are connected to lipid molecules. While the peripheral membrane proteins, they don't interact with the hydrophobic core, the tails of the phospholipids. They don't interact at all. And they bound to the membrane indirectly by interaction with integral protein. So you can see that the peripheral protein can bind to the integral protein indirectly, okay, or directly by interaction with lipid head groups. These peripheral proteins are either localized on the cytosolic surface or on the exoplasmic surface of the plasma membrane. And you can see here from the figure, if you enlarge a part of the EM micrograph we have just seen in previous slide, if you enlarge a part of it, you can see in a diagram, it consists of a lipid bilayer. You can see the lipid bilayer, in which let's have a pointer. You can see it consists of lipid bilayer where the head groups are oriented to the outside, and there is inner, for the phospholipid bilayer, there is inner hydrophobic core. And you can see here the different three categories of proteins, which are the integral protein, the transmembrane protein, which is either like an enzyme or something like this, or having a channel. And these two kinds, which are transmembrane, are called integral protein. You can see as well inner peripheral protein, and one of its function is a connection with part of the cytoskeleton. And you can see as well outer peripheral protein, and another type of the outer peripheral protein, which is connected to the carbohydrate molecule. That's why it's called the glycosylated. That's another type of protein. And you will note that this glycosylation of these carbohydrates bound to the protein is very important. Is a very important mechanism in forming a structure that we'll know later is called the glycocalyx, which is very important in the recognition of the cell, of any cell type. This another simplified figure, we can see the different types of protein and as well the phospholipid bilayer. So you can see the phospholipid bilayer separating from the exterior from the interior, and you can see how the different types of protein are differentiated. You can see the integral protein, transmembrane. You can see outer peripheral, inner peripheral. You can see the lipid anchored protein either from outside or from inside, and they got connected to special phospholipid molecule. You can see that the peripheral, internal peripheral, can bound to the cytoskeleton, while the external peripheral, it can be glycosylated, and these are which appear like fork in structure. These are the carbohydrate chains. And you can see that this integral, either integral or peripheral protein, or even the lipid anchored protein, can bound to the extracellular matrix, which actually surrounding the cells. So let's see some examples. Okay. Let's see some example of membrane embedded alpha helix, which are the primary structure, secondary structure in most transmembrane proteins. And we'll talk about three examples. Okay. You need to be familiar that protein actually in their secondary structure can appear in the form of alpha helix or a beta chain. We understand now how these proteins got embedded inside the biomembranes. And we'll talk about three examples. So this repertoire of folded structure in integral membranes protein is quite limited, and the hydrophobic alpha helix actually predominate. Okay. So this alpha helical domain, which is a part of integral protein, can span and embedded in membrane by hydrophobic interaction with specific lipids. And this ionic interaction with the polar head, they can do ionic interaction with polar heads, polar head groups of the phospholipids. The first example is glycophorin A. So glycophorin A is actually an example of single pass transmembrane protein. What's the meaning of single pass? That means that this type of protein can only crosses the membrane just once. And actually, it's a major protein in the red blood cells plasma membrane, and it contains only one membrane spanning alpha helix that is consisting of 23 residue or amino acids. The helix of one molecule associates with the helix in another to form a structure that is very peculiar, which is called coil-coil dimer. And many cell surface receptors are activated actually by dimerization. So that's why two molecules, the helix, the alpha helix of two molecules of that protein bind together in the form of coil-coil dimerization, which is a very crucial in the activation of this type of integral protein or this type of receptors. And you can see here a figure of glycophorin A. The first thing you need to notice that is actually it is formed of two molecules. And the part that is spanning inside the phospholipid bilayer, actually it is the alpha helix. This is two alpha helix of two different molecules. And this is a phospholipid bilayer. And for sure it is integral protein, transmembrane protein, because you can see that each molecule only have a single pass, one time pass through the membrane. And it have an extracellular domain, part of the protein, and they have a cytosolic domain. And there is a dimerization, and you can see attached. Even they managed to know the sequence of amino acid from 73, the amino acid number 73 to amino acid 96. Actually, these are the sequence that spans in the phospholipid bilayer. You can see as well that the extracellular domain is glycosylated, and they have carbohydrate side chains on the serine, threonine, and asparagine residues of the extracellular domain. And you can see as well, these blue tagged amino acids, these are positively charged arginine and lysine residue. These amino acids are very important because they are positively charged and they can bind to the negatively charged phospholipid bilayer. That's why it can anchor this glycophorin A in the membrane. It's a very crucial for the stability of that protein and the anchoring of this protein into the plasma membrane. The second example of the integral protein, actually another famous protein, which is GPCR, G protein coupled receptor. And it's another example of the multi-pass transmembrane protein. This actually is a receptor, G protein coupled receptor. And it's one of the 150 integral protein multipaths that have been identified. And one of the main difference between this protein and the other protein, Glycophorin A, that it have 7 membrane spanning alpha helix. The first example, which was having only 1 membrane spanning alpha helix. So you can see here, it transverse membrane 7 times, H1, H2 helix, that's mean alpha helix, H1, 2, 3, 4, up till 7. 1, 2, 3, 4, 5, 6, 7. So actually it's a very huge molecule. And this actually is the shape, the actual shape in a membrane. These are the alpha helix, it appears like coils. So you can see that there are 7 transmembrane alpha helix. And you can see that there are 4 extracellular segments from E1 to E4. And there are as well 4 cytosolic segments. And you can see that this part, C3 and C4, actually this part is used to bind to another very important protein, which shares its part and play an important role in the process of cell signaling, which is the G protein. So that's why this receptor is called G protein coupled receptor. That means that this receptor, once activated by the signal from the outside, it conveys the signal through C3 and C4 to activate another important protein, which is the G protein. And actually G protein binds to that receptor through the C3 and C4 cytosolic segments. And that's why it's called C, cytosolic, while the other one is called E, which is extracellular. Let's talk about a third example, which is an ion channel. For the ion channel, They are as well multi-pass transmembrane protein. One of the good examples is the tetrameric ion channel, which is tetrameric protein. What's the meaning of tetrameric? That's mean it's a huge structure that is composed of four molecules to form a big structure, tetrameric. And we will take an example of that, the crystal structure of the potassium channel. Each of these four subunits in the tetrameric proteins has a pair of membrane-spanning helix that bundle with helix of other subunit, forming a central channel. So you have four molecules. Each of these four molecules has a pair of alpha helix, and so you have like now 8 alpha helix. They are connected to each other in the form of a circle to form a central channel where potassium can move. So in many ion channels, the external factors, which is actually the ligand or the voltage or the mechanical strain, regulate the ion flow across the bilayer membrane by reorienting the helix. So in other words, as you already know now, that the helix, the alpha helix, actually it is a part of the membrane, the part of the protein which transverbs the membrane. The reorienting of this helix in an ion channel will close and open the ion channel to enable the flow of the ions. So in this figure, you can see this actually is a one molecule, single subunit of the tetrameric. And you can see it is alpha, it have two alpha helix. When combined, and when you look at from the side view, you can see that there is a channel is formed for the potassium ion to form. If you look closely from the top of this ion channel, you can see actually it is composed of 4 different molecules. Each of them have 2 alpha helix. And actually this actually the potassium channel from the bacteria, the bacterium Streptomyces, okay? So as we said, it's a tetrameric protein for this potassium ion channel, and each subunit of this, the tetrameric protein, have 2 alpha helix, an inner helix and an outer helix. And this inner and outer helix, they found that it is conserved. What's meaning of conserved? Conserved means that the amino acid sequence of this alpha helix is conserved. It is actually the same from the primitive organism up till to higher organisms, even humans. So now we'll talk about different type of transmembrane protein, but in this case here, it's not alpha helix, it's a multiple beta strands. One of the famous example is porins, and it form like membrane spanning barrels, okay? And these porins are transmembrane proteins that differs completely from other integral protein. It is still integral protein, but why it's different? Because actually the part that is spanning inside the plasma membrane, actually it's a beta chain, not alpha helix. These porins are found in the outer membrane of gram-negative bacteria like E. coli, and the outer membrane as well of mitochondria and chloroplasts. In E. coli, this beta porin permits the uptake of nutrients and disposal of waste product. So actually, it's like a doorway that allows the entrance of nutrients and the expel of the waste products. And these porins provide channels for the passage of the disaccharide and other small molecules. The X-ray crystallography, which actually technique used to understand the structure of biological molecules, have revealed that porins are trimers of identical units, subunits. Each subunit of these trimers consists of 16 beta strands. 16 beta strands form like a barrel-shaped structure where the bore is in the center. Let's see here a model. So actually what you are seeing here actually it is just one subunit. And as you understand, we have just said that the porine is consists of trimer. So you are seeing here only one subunit. And if you count these beta sheets, you will find that it is 16. So actually, it shows that it is integral protein because it's a transmembrane protein. It have a cytoplasmic phase part, and it have an exoplasmic part, and it have a transmembrane. But the difference here that it is multipass. You have beta sheets transversing. You have 16 for each subunit, and this actually the structure model of one subunit of porine found in the E. coli outer membrane. If we look from the top, we can see from the top or from the outside of the bacteria, you can see the three different subunits. And you can understand that these 16 beta sheets that are transversing in the membrane, when aligned, they make a pore in the middle. So what about aquaporine? Actually, aquaporine works on the same principle, but from the name implies it's only purely responsible for the movement of water molecules. So actually, it is water channel in the plasma membrane of animal cells. And this aquaporine is a multiple transmembrane alpha helix of 4 identical transmembrane helix. It differs structurally from the porins. Okay, it's totally different because actually it is the transmembrane part of the protein is formed of alpha helix, while in the porine it is formed of beta sheets. So it differs. This aquaporine differ structurally from the porins as well as functionally, because it's only transport water molecule. It mediates the transport of a single water molecule, namely water. So you need to understand the major difference between the aquaporine and the porine. So porine is responsible for the movement of nutrients as well as waste product in the bacteria. But aquaporine is exclusively for water molecules. And you need to understand that aquaporine actually exists in humans. And actually, this aquaporine exists kidney, in the kidney of humans, in the membrane of tubule, the renal tubules of the nephron, because they are responsible for the process of reabsorption of water through the process of the formation of urine. Okay, so let's see now. Actually, this is the structure of the just hold on. The structure of the aquaporine, as we have just said, it's a 4 identical transmembrane alpha helix, and it's multiple. So you can see here, and this is the 3D structure of it. Okay, and you can see in the middle there is a pore. Okay, so let's talk about the lipid binding motifs. Lipid binding motif that help some of the peripheral proteins to the membrane. So the help in binding of this peripheral protein to membrane. So these proteins, some protein have a kind of domain or motif, which is called Blackstone homology motif or domain. Usually, we just have the initial pH. So this binds 2 types of phosphorylated phosphatidylinositol, which is BIB2 and BIB3. Actually, the difference is 1 phosphate. So what's BIB2 and BIB3? Actually, it's 2 types of phospholipids that can bind to any protein which have the domain pH domain. So you can see here, this is the phospholipid bilayer, And these two types of molecules, which is BIP2, and this is PIP3, so the difference only is one phosphate. And it is a special type of a phospholipid that exists in the bilipid bilayer. And you can see that any protein have part of this protein, we call it domain or motif, that is have a specific sequence that scientists termed the name a pH or Blackstone homology domain. It can bind to either the diphosphorylated phospholipid or the triphosphorylated phospholipid. So what is that pH? pH initially recognized in protein in platelets, which is called Blackstone, and this pH high frequency indicate that the protein can be localized to the membrane surface, and carry important, very important. Any protein that have this pH domain will carry a very important function in the process of signaling. What about C2 domain? It's another type of domain, and it is common lipid binding motif, and originally discovered in a kinase which is called protein kinase C, BKC. And these proteins membrane target domains for various kinases like PI3 kinase, phosphatase like PTEN, and phospholipase. To summarize this slide, actually it's saying that, if you remember that we have said that there are types of protein that can be anchored to lipid. So actually we understand now any protein which have a specific sequence, a termed domain like the pH sequence or the pH domain or the CT domain, have the ability to bind to a lipid molecule in the plasma membrane. So the plasma membrane actually has many functions that is related to the cell. And it has many functions common to all cells. Its lipid composition of the plasma membrane determine its physical characteristics. And its protein composition is primarily responsible for the function of that and the properties, the functional properties of that plasma membrane. So the major function, actually it acts as a permeability barrier. The specific membrane transport protein permit the passage of nutrients into the cells and the metabolic waste outside. It maintains the proper ionic composition and the pH of the cytosol, which is around 7.2. The PM is the plasma membrane is highly permeable to water, but poorly permeable to salts and small molecules such as sugars and amino acids. Owing to the osmosis, water moves across from solution of a low solute, which is high water, into solute concentrated one, which is a low water concentration, until the total solid concentration will be equal on both sides. And the proteins of the plasma membrane and the glycolipids form specific junctions between cells to strengthen the tissue and to allow the exchange of these metabolites between cells. While the receptor, which is a type of integral protein of plasma membrane, binds the specific signaling molecule, which is either hormones, growth factors, neurotransmitter, and this will lead to various cellular responses. The peripheral cytosolic protein that are recruited to the membrane surface function as enzymes, intracellular signaling transducer, structure proteins for stabilizing the membrane. So in these two slides, we summarized the plasma membrane major function.